Hello everyone, welcome back to the channel. Today we are going to look at how you can control the mind with lasers. Optogenetics is a neuroscience technique in which you can manipulate neurons, a type of brain cell, with light. Neurons communicate with each other via electrochemical impulses, and this communication allows us to learn, remember, speak, move, and in general, think. But how can we manipulate neurons with light? As the word optogenetics indicates, using genetic methods. First, we need an algae. The algae can detect light of a particular color. It's a unicellular organism that will move away or towards light when it detects it. How does it manage to detect and react to light, though? Well, this green algae has in its cell light-sensitive channels, which are our detectors. Once the channels open, the late sodium ions go through, which activate the cell. This is similar to the way in which neurons are activated or deactivated. They do it thanks to channels in the cell's membrane, which open and close. In our brain, these channels only usually open when they detect a neurotransmitter, which is a chemical substance secreted by other neurons. Once open, the sodium ions go through and the neuron sends an electrical impulse to communicate with the other neurons. So here we have our light detecting channel and a response. Now, how can we insert these channels in our neurons? Using a virus. We can take the channel's DNA code and put it in a virus. The virus is then injected in the brain and infects the neurons. The infected neurons then express the algae's channel and then are able to respond to light. So we can use LEDs or lasers to activate these channels by implanting an optic fiber in the brain. Depending on the biological detector we use and the light color, we can activate or inactivate only certain neurons and not the rest. For example, with channel rhodopsin and a blue laser, we activate, and with halo and yellow, we inactivate. As the technique is very precise, it does not infect all the brain, and only a type of neuron where the injection took place may express the light channels. Now to the cool stuff. How these lasers affect behavior. In a very interesting study, scientists investigated what happens when we activate a memory in mice. The mice learned to dislike a box which had metal bars, but really enjoyed being placed in another box that had different floor and walls. Then the animals were placed back in the box they liked the following day, but this time the cells which were active in the box with the bars were stimulated with light. And then the mice started disliking their preferred box as well. They were implanted with a false memory. This kind of studies can help us understand how memories are formed and how we remember them. We know that sometimes neural communication fails, like in Parkinson's disease where patients have a movement disorder, or Alzheimer's where memory is lost. Optogenetics is more precise than electrical stimulation with electrodes and faster than drugs. Perhaps this technique may help reestablish communication between certain neurons and, for example, improve memory. So this is all for today. I hope you liked the video and I'll see you next time. Bye bye!